Hospitals around the world are currently fighting a losing battle against Acinetobacter baumannii. It is a highly aggressive pathogen that thrives in intensive care units, causing lethal infections that shrug off our best medical interventions. For decades, doctors relied on a standard arsenal of broad-spectrum antibiotics to clear out bacterial invaders. But against this specific strain, those conventional drugs are increasingly useless. To understand why, we have to look at the bacteria in its natural or wild state. Acinetobacter secretes complex chains of sugar molecules that weave together to form a dense outer layer known as a polysaccharide capsule. This capsule functions like a biological riot shield. When hospital-grade antibiotics or the destructive proteins from our own immune system try to breach the cell wall, they hit this thick sugar coating and harmlessly deflect away. As long as the pathogen remains wrapped in this armor, the infection spreads, and doctors are left with no effective way to stop it. Since chemical drugs can't breach the barrier, researchers are turning to biology. Specifically, they are isolating natural predators that have evolved over billions of years to hunt down and kill bacteria. These images show two distinct bacteriophages, known as FG02 and CO01. Researchers at Monash University recently isolated these specific viruses from raw sewage because they displayed a unique ability to target the Acinetobacter superbug. What makes these phages effective is exactly what makes the bacteria so dangerous. The virus uses the thick polysaccharide capsule as its docking station. It locks directly onto the sugar molecules, using the bacteria's own shield as an anchor point. Once firmly attached, the phage injects its genetic payload straight through the bacterial wall. The virus rapidly multiplies inside until the pressure builds, causing the armored bacterium to burst open and die. The medical implications here are severe for the pathogen. Its greatest evolutionary advantage, the impenetrable capsule, is the exact feature the virus tracks to kill it. Faced with a targeted viral assault, bacterial colonies don't just wait to be wiped out. As they divide and replicate, they undergo rapid random mutations, desperately searching for a biological workaround to survive the phages. To escape FG02 and CO01, the surviving bacteria employ an extreme survival tactic. They introduce a single nucleotide deletion into their own DNA, intentionally breaking two specific genes known as GTR29 and GPI. These genes act as the instruction manuals for building the protective outer layer. By disabling them, the mutant bacteria completely halts the production of its polysaccharide capsule. These electron microscope images reveal the physical cost of that survival strategy. On the left, the wild-type strain retains its thick, highly textured surface. On the right, the new capsule-deficient mutant is completely smooth and naked, rendering it entirely invisible to the attacking phages. The bacteria successfully evaded the viral threat, but the cost of that evasion was the permanent destruction of their own biological armor. This leaves the hospital with a seemingly new problem, a growing population of mutant bacteria that are entirely resistant to our phage therapy. But without their protective shell, these mutants are exposed. The old chemical treatments that used to bounce right off the cell wall can now penetrate the naked bacteria. In medical terms, the strain has been resensitized. This table tracks the minimum amount of antibiotic needed to stop the bacteria. Look at standard beta-lactam drugs like ceftazidime. The wild bacteria required massive doses, earning an R for resistant. The naked mutants required 16 times less, flipping directly to S for sensitive. This chart shows how the human immune system reacts. The red lines track the wild strain easily surviving in human blood serum, but the blue dashed lines show the mutant strain plummeting to 0 in under 45 minutes, utterly eradicated by natural complement proteins. To verify this mechanism works in living tissue, researchers tested the therapy in mice infected with the superbug. When they introduced the specific phages, the bacterial load in the solid organs and blood plummeted by orders of magnitude. Throughout the treatment, the virus is actively replicated at the site of the infection, multiplying their numbers to continuously hunt down the remaining wild-type pathogens. The clinical battle against superbugs often feels like a losing arms race, but by using carefully selected bacteriophages, we can trap pathogens in an evolutionary catch-22. We force them to make a choice: retain their armor and be destroyed by the virus, or shed their armor to survive. rendering themselves highly vulnerable to the human immune system and the standard antibiotics we already have on the shelf.